you. All right, welcome to those of you who are here in person and those of you who are here virtually. My name is Dr. Denise Dupra, and I am the Associate Director of Education for the Center for Individualized Medicine here in Rochester. I would like to welcome you to the Center for Individualized Medicine Grand Rounds. The CIM Grand Rounds lecture series is designed to highlight the latest in scientific discovery and innovation and demonstrate how individualized medicine is being translated into practice to meet current and future patient needs. It is my pleasure to introduce today's presenter. Dr. Stephen Boppart is the Director of Interdisciplinary Health Sciences Institute and Professor and Granger Distinguished Chair in Engineering at the University of Illinois, Urbana-Champaign. He holds joint appointments in the departments of electrical and computer engineering and bioengineering. He is also co-director of the Mayo Clinic and Illinois Alliance for Technology-Based Healthcare. Dr. Boppart leads multiple major initiatives, including as director of the NIH/NIBIB P41 Center for Label-Free Imaging and Multiscale Biophotonics and the GSK Center for Optical Molecular Imaging and the Biophotonics Imaging Laboratory at the Beckman Institute for Advanced Science and Technology. He is also affiliated with the Cancer Center at Illinois. Today's presentation is titled, Imaging Life with Light, Not Labels. Today, he will lead us in exploring the future of biomedical imaging, how innovative, label-free optical imaging technologies are enabling us to visualize living cells and tissues based on their unique intrinsic optical structures and signatures. These advancements move beyond traditional chemical dyes and probes, offering a non-invasive way to study biology without disrupting it. From creating stain-free, slide-free digital histopathology images to capturing real-time optical biopsies. During surgery, these tools are transforming clinical workflows and diagnostics. The presentation will highlight how these technologies are unlocking new possibilities in AI-driven technologies, diagnostics, biomarker discovery, and spatiotemporal profiling of the tissue and tumor microenvironments, paving the way for more precise, real-time insights into health and disease. Throughout this talk, for those online, please use the Q&A in the Zoom to submit your questions. We have planned time for Q&A at the end of the session. For those in person, we will pass the mic around the room. For those who have not yet claimed continuing education credit, the code is BOPART, all caps. Please welcome, with great pleasure, Dr. Stephen Boppart.
Well, thank you very much for the warm introduction and just really the opportunity to be here. I'm really thrilled to be back here in Rochester and to interact with so many of you, a longtime colleagues here as well as new faces. And as was mentioned, I'm really looking forward to sharing with you some of this technology and these ideas of how we can use imaging with optics and with light to really explore a lot of these new markers, signatures from tissue, and use those for many different applications in medicine. Just a few disclosures to start with here. Federal funding, some support from GSK. Also, I've been involved in a number of startup companies, and all of our animal and human studies were done under approved protocols. If we think about biomedical imaging, and let me see if I can move this in some sense. I don't know. There, maybe that at least gets that out of the way a little bit better. If we think about biomedical imaging across multiple scales, right, we're interested in structure, interested in function as well, again, across all these scales. And what comes to mind is really a lot of these radiological imaging modalities, the CT, the MRI, the X-ray, the ultrasound at these scales. But it really is light and biophotonics that allows us to look and probe at that cellular molecular level where many of these diseases start. And that's what has motivated us to develop these types of imaging modalities. Now, as was mentioned in the introduction too, I'm also director of a national NIH center called the Center for Label-Free Imaging and Multi-Scale Biophotonics, or CLIME. And our charge there is to use these label-free technologies and really move from the cellular level all the way up to the clinic. So cell to clinic, and thinking about the cell dynamics, the digital pathology, how we can then use this to look at in vivo physiology and ultimately clinical imaging applications. And so we've been thinking about how to do this too. So we're not limited to just at that small microscopic scale. Ultimately, what we're talking about is this term we like to say, optical biopsy of tissue. I think we're all familiar with the physical biopsy is, where taking out that tissue specimen, fixing it, processing it, sectioning it, staining it, looking under that microscope for that diagnosis. But the idea is, can we actually think about how these optical technologies can allow us to visualize that cellular structure even during procedures, surgical procedures, and not only look at the structures, but also some of the function, the movement, the dynamics of these cells. So what we're thinking about is how we can change this paradigm for when, where, and how we assess tissue microscopically with light. Well, histopathology is a gold standard, right? It's hundreds of years old, historical. It's, however, very slow. It's time consuming. It's labor intensive. I know Mayo is a leader in terms of the frozen sectioning in pathology, and that's really accelerated and enabled this type of microscopic view in a much faster time scale. But historically, this type of technology is used really for only ex vivo specimens. In the end, it's the images that the pathologist used to make that diagnosis. And so if we can generate those images in such equivalent way, then we can open up all these other opportunities, even begin to do this in fresh, unstained tissues, or even in vivo in many cases. So that's what's driving us. And if we look back at the many different modalities, almost any biomedical imaging modality has contrast agents that are used to enhance structures, to identify features. So using X-rays and in X-ray CT, there's various agents. In MRI, there's gadolinium-based agents or nanoparticles. In nuclear imaging, these radionucleotides. Ultrasound uses microbubbles. Wide field optical imaging, very often approved dyes are injected or administered topically to highlight different structures. And then at the microscopy histology scale, there's the stains, or there's the quantum dots, or GFP, which are genetically expressed fluorescent probes that can be used for imaging. And so this type of visualization certainly enhances our ability to see structures that we're interested in. And I always like to say, sometimes when you use contrast agents, those are helpful, but it's only to image what you're looking for. And what else maybe are you missing that's not being labeled? So label-free imaging begins to afford some of this ability to look at other structures as well in new ways. And there's many different advantages. So we're going after intrinsic signals that we can extract from the tissue, even fresh, in vivo tissue, new forms of contrast, which can yield new features or biomarkers, a structural, functional, metabolic imaging, as I'll show. And there's really no confounding factors associated with targeting. Oftentimes, when you add a contrast agent that's targeted, you want it to go to your target, but sometimes it labels off-target structures, or maybe it's an incomplete targeting, and you have those questions as well. And anytime we add a dye or an agent or a drug, it's going to raise questions of toxicity, require FDA approval, and an additional level of safety and approval for those dyes. Label-free, though, we can highly multiplex these signals in different ways. It also allows us to look at new physics of how those optical signatures are being generated. And you'll see examples of how we can use this to look at spatial biology, and this feeds directly into AI, which is excellent at pattern analysis. Now, the disadvantages of this label-free approach is that, of course, we have a relatively limited source of contrast, and we have a lower specificity because, again, we're not specifically targeting a particular dye or contrast agent. We are putting in optical energy, but we can control that. It's safe. The signals we get back are often weak, and we have to often use AI to look at extracting some of those signals. But hopefully what I'll convince you is that there's an extreme benefit of considering these label-free approaches. Now I'm going to get a little more technical. One of the advantages that we have here is that we can manipulate light in different ways. It's not just illuminating a sample and looking at what comes back, but what's important here is that we can actually engineer the type of light that we use to illuminate the tissue with. So my lab does a lot of work in optical engineering and science to design new light sources, to be able to generate new wavelengths and new forms of light, even very short optical pulses. And so when we have a very short optical pulse that is impinging on the tissue, it can excite many different wavelengths and signals that come back. And we do this, you can see in the lower right here, we can pump these optical fibers, and it's these special fibers that broaden the spectrum, many, many colors that start to come out. So this is what we call spectral and temporal tailoring of the light. And so the idea is this: instead of taking out the tissue, fixing it, sectioning it so thin that you lose contrast, you put the section on a slide and then you add dyes back to give you the contrast. Instead of using that white light microscope, we will illuminate the tissue with many different wavelengths and many different properties of light. And each of those produces these intrinsic signatures that come back and provide new forms of contrast. So it's a way of programming the light to give us the new contrast. So instead of that stained H&E section, what we get are images that are stain-free, label-free. So this is just cycling through. It's fresh rat tissue that is being illuminated with different wavelengths and forms of light, and we're getting all these types of contrast back inherently from the tissue. And I'll go through these mechanisms. But this we can do in minutes as opposed to hours or days often. Now, along the way, we can discover new biomarkers, and those little blue dots I'll come back to, those are extracellular vesicles. And we are the first to really be able to identify these label-free in situ. And of course, there's a big interest now in trying to understand how cells communicate through these extracellular vesicles. So if we put these all together, we get images like this. This is a composite of multiple different modalities of imaging, and this is all label-free. So this was a fresh, resected, unstained, unprocessed tissue, and we can see structures, blood vessels, we can see collagen, the larger adipocytes are shown there. Even the little red blood cells, the biconcave disc, is seen there in the blood vessel. And again, all label-free, all just within seconds to minutes of this type of imaging. This is what we like to call true colors of cancer. This is a tumor specimen, but I give a public talk where I talk about these colors. Now, the colors that you're seeing, of course, we can arbitrarily assign colors, but my graduate student said, well, why don't we just assign the color of the wavelength that's truly coming back? So the colors you see in this image is that wavelength or color that's coming back from the tissue during our imaging. It's the true colors. And we just, these obviously are too weak signals to see with your naked eye, but our systems can pick this up and image this way. Okay, well, how do we do this? All of these are generated through nonlinear interactions. Nonlinear interactions are when multiple photons of light interact with a molecule in the tissue, and in some different ways will produce other photons of light, so other colors that may come back. We're all familiar with fluorescence. So we can generate fluorescence with this multi-photon nonlinear approach if we use 2 photons that arrive at the same time and place, or 3 photons that arrive at the same time and place. And there's examples here. So that 3-photon fluorescence that we're detecting is autofluorescence coming from NADPH, and that's literally everywhere, involved in metabolism. The 2-photon autofluorescence is coming from FAD, which also autofluoresces. Now we have harmonic generation. This is a different form of these nonlinear interactions, but it's more about the structural features in the tissue. So second harmonic generation comes from collagen and elastin. Third harmonic is really good at identifying interfaces between, say, lipids and aqueous compartments. And then finally, CARS and SRS are vibrational techniques that look at the composition, molecular composition, lipids or proteins. Now, nature has really given us a palette of different colors, different signatures, different endogenous optical signatures that we can extract from this. And this is a process that we use, we exploit this. And in fact, we call this technique SLAM microscopy. SLAM stands for simultaneous, label-free, autofluorescent, multi-harmonic microscopy. And the important point to mention is that this all happens simultaneously. So we can put in a single optical pulse that's maybe femtoseconds in duration and collect then all of this signal that's coming back. So a few more technical slides. So with SLAM microscopy, we did a lot of engineering to figure out what is the ideal wavelength that we want to excite all of these processes with. And that's what we're calling this optimal window around 1100 nanometers. You can see below that, you can see the sort of optical spectrum that our laser generates. And then we can select which regions within that spectrum we want to excite the tissue with. Now, we chose that optimal window because then all of these signals coming back, we can separate cleanly the best we can with different filters. So we get all these back, and we can implement this in a platform, as you can see here. Essentially, it's a platform where we're collecting light in the epi direction, so the backward direction. This is important because, well, those signals are weak, but it allows us then to image thick tissue. And we can, take our microscope objective, apply it to the tissue, and collect the signals back that way. It's not in a transmission mode like a lot of microscopes operate in. And then you'll see there's multiple detectors to separate and detect all of these channels and signals coming back. Well, with this, we can do this quickly too. So these are some example images. In the top is showing a fresh brain slice from an AD mouse, an Alzheimer's disease model mouse. And what you're seeing are plaques, AD plaques, AB plaques of amyloid beta. And again, the unique contrast we can see with these. Below, you're seeing blood flow in this in vivo mouse brain. Now, I have to admit, we did inject a dye into the vascular system here, but not for imaging. But what you're seeing are gaps where red blood cells are passing and blocking out the dye. And then on the right, you can see these are individual spermatozoa within, even in vivo. So being able to image those and their dynamics very quickly at very high frame rates. So not only can we collect this label free, but we can also collect it pretty quickly to capture dynamic events. Here's a few more examples, starting from the left. So third harmonic, again, looking at lipid aqueous interfaces. This is just the skin, inner skin of the mouse, where we see blood flow, we see some other structures. C. elegans is in the next column. So 3-photon autofluorescence, that's the NADPH autofluorescence coming from this live and twitching little worm. In this label-free approach, we can look at developing embryos in these bovine embryonic cells. And then on the right, of course, just looking at individual red blood cells through this amniotic membrane. So again, examples of label-free dynamic imaging, just exploiting this label-free contrast. Okay, coming back to the in vivo rat mammary tumor, we can see structures like this when we start putting them all together. Again, the green is collagen, the blue are different cells. We've zoomed into a couple of them here. Endothelial cells. You'll see that, you'll see here, there are cells with yellow cytoplasm and some with blue cytoplasm. That's based on the metabolic activity we're seeing from those individual cells. So again, and I'll touch more on that. This is one of my favorite images. It's an in vivo neurovascular bundle. So you see this large vein structure running down the middle. There's an artery that's next to it. The magenta is coming from the myelin wrapped around the axons in that nerve bundle. And then you've got adipocytes surrounding that. This is in a bed of collagen. And then those yellow are fibroblasts that we see. Now, this is in vivo, but it was a mosaic. So we stepped over multiple frames to capture that. If we took a single frame and looked at it over time, we can start to see, the dynamics. So this is a normal exposed rat mammary tissue. You can see a vessel with red blood cells flowing through. Here's another example. So pretty linear flow. You can see some changes in the flow rates. Mostly you're just seeing red blood cells here, but I want you to contrast that to some other examples. Well, here, if we look and zoom into some of this blood flow, we can watch and even see cells that are extravasating, intravasating. You'll see a lymphocyte there rolling by. We can track the trajectory of that. Here's another example. And this one, I think it's extravasating. So rolling there and tracking its position. So seeing some of these dynamics that are happening on very fast timescales. But this was actually an exposed rat mammary tumor at the periphery. And now all of a sudden you see that the vasculature is very, distorted. We see a lot of activity outside of the vasculature. So these are migrating, we don't quite know, tumor cells, lymphocytes, macrophages, immune cells. You can see we're tracking these. This is over 70 minutes, a time-lapse image, taking a frame every 2 seconds. And we can see all these dynamics. So the point here is there's a lot of activity going on. There's a lot of information here that we can extract. And so if we look at these more quantitatively, we can start to see general activity of these cell dynamics. We see that from the start to the finish, there were a lot of these clustering of these cells that were migrating around the bend of this vessel. And in fact, we can track the trajectory of each one of those cells. And they have pretty typical similar patterns. They all have this same, the D is directionality. It's a time, like a burst of activity, and at a fairly uniform velocity. So that's a whole separate class of cells. There are other cells that migrated long distances or short distances and had very different dynamic characteristics. This comes into the sensitivity question. We're seeing these differences in cell dynamics and behavior within in vivo here, label free, but we don't exactly know yet what types of cells these are. And we're trying to figure out, we can go in and label all of this technique. These techniques can still use labeled approaches. And we can go in and label these to determine exactly what cell types. Once we know what the label-free signature is of a particular cell type, we don't need the label anymore, and we can track those. So the other point I want to make is that with these measurements, we can then begin to assess the metabolic activity. So it's very often that we define this optical redox ratio, which is a ratio between that autofluorescence of the NADPH and FAD. And if we apply this analysis to these cells, we saw something really interesting. All those blue cells were the ones that had migrated to the site, and we compared them to the cells that were migrating in the actively moving. Surprisingly, though, again, this lower ratio means more metabolic activity because the NADPH is higher. The clustered cells were more metabolically active than the migrating cells. And we think it's because they're upregulating gene expression, they're doing something else genetically. But that was a surprise to us. And we wanted to validate this metabolic imaging using a much more controlled type of experiment. So these are macrophages that we can activate with interferon gamma or LPS. And what you're seeing in blue around the periphery of those nuclei are the perinuclear mitochondria. And so we can actually track when these are activated, those light up, right? There's more activity, autofluorescence coming from those perinuclear mitochondria, just confirming what we're seeing here. Now, the other important point I want to reemphasize is that all of this data collection is happening simultaneously. So we did this study. That compared what if someone just captured these images, these channels, say a few seconds later, or maybe there was a slight misalignment, the cell moved, for example. What would that change? And what we found is it does change this measurement of metabolism dramatically. So if we perfectly temporally and spatially co-register, then we see, this type of image up here. And when you start to see the blue and the red, that means that redox ratio is changing, right? It's different. And so it just validates why this type of system with simultaneous acquisition is so important. Many other groups do multimodal approaches, but if there's a time or a space difference, you lose that information. It's no longer accurate. We also validated our images as best we could with, stained H&E, immunohistochemistry, many different processes. And you'll see that there's a lot of structural similarities between the two, but we're seeing things in our imaging that don't show up in H&E or in histology. We're seeing new features because we're not necessarily looking for a labeled structure. Some of those include extracellular vesicles. And in fact, extracellular vesicles are washed out in the histology procedure, but they're able to be retained and imaged in vivo. Knowing this type of assignment, this is ongoing because we're still trying to associate our signals and images with the labeled known structures. But this is going to allow us, I think, to discover new markers. Now, I think we can all admit that AI and deep learning are just changing everything that we do. And is this the final disease diagnosis? It certainly is advancing biomedical imaging in so many ways, and optical imaging, the pathology, the imaging that we do. The reason is because, again, AI is good at finding patterns in data that we can't see or interpret with our own senses. And so when you have images that are multidimensional, you all of a sudden have so many more patterns than what you can really appreciate. So it's ideal for this, and even more ideal for this multimodal approach. So we not only have a single image, we have all these different channels, and there's patterns in relationships between the multiple channels, and we can take advantage of that. So one way we can use AI is to segment out our images and identify cell types that might be present. We can come back to this image, and using supervised learning approaches, we had people identify different cell types in the images and associate those to train this model. And the output of this model could tell us, in a very general sense, where are the tumor cells, the stromal cells, the leukocytes, macrophages, and give us a pie chart of that distribution. So now, and we can zoom in here, there's even the little dots, which are the vesicles. But now we can give this model any one of our images that we could collect, and just instantaneously be able to identify the cell populations and their distribution. This is our spatial biology, right, of identifying where those cell types are. So this is largely collagen with a few of those fibroblasts. So it's identifying all those stromal cells. We can use this then to what we call deep profiling of the tumor microenvironment. And again, move away from the labeled spatial biology and omics to think about label-free spatial biology, spatial omics. And not only the spatial part of this, but because we don't need to fix and stain and kill the samples, we can now have label-free temporal biology, temporal omics in the end, okay? So we developed this type of model here where, again, we could feed in these images, these multimodal images, the multiple channels, and get out not only the quantitative metrics of numbers of cells, the size of cells, their signatures, but also their relationships in space. And this is just some examples of how we can do that. But once you characterize your image like this, you can then begin to look at those cell-to-cell relationships, that spatial positioning of those. And that spatial arrangement, too, has clinical significance, diagnostic significance. So this is just meant to kind of show you there's lots of properties that we can measure from these cells, from these images. Again, the morphology, the metabolism, a lot of these different channels, and look at differences between the cancer tissues and the normal tissues. In addition, we can develop this concurrence mapping, so we can look to see when this cell is present, what's its neighbors, right? What neighbors are present? When there's extracellular vesicles around, what cells are they next to? And what does that spatial arrangement look like, again, in states of control or health in cancer? This is just graphically to show some of those results. First off, you can see that the cellular distribution obviously is different between control and cancer. A lot of these concurrence matrices show differences, but then across the bottom, the x-axis is that optical redox ratio, sort of the metabolism. And then the third harmonic, again, is the structural features. And you can see clear differences in these distributions between the cancer and the control tissues. So again, just something we've really only started to demonstrate and tap the surface of, and a lot more work to do there. But it's headed toward this idea of label-free spatial temporal biology and omics. So what's being shown here is really the standard of care, or standard of procedure, where we again go back to the sectioned, stained slides of tissue sections. And a lot of these spatial biology systems will go through cyclically and apply an antibody-based stain or dye, take an image, and then cyclically wash that off and restain, and do this, tens of times. And you get these beautiful images that are labeled with all these different probes. Well, the technology kind of stops there. This can take days to be able to generate a single image. And so it's that same challenge that we were trying to address with histology, we're now trying to address in the spatial biology. So instead of doing all that, can we use this type of label-free, very fast system to generate these multidimensional images? Now, you saw all the different contrasts we were generating. What we don't know, though, again, is the correlations between these two. So that's where AI comes in, and this concept of image to image translation. So what we're doing now is we're imaging tissue fresh, we're doing the histology, we're doing the current standard of practice, and then we're going to use AI to compare the two. And if we can develop enough correlations between our label-free images and the stained spatial biology, spatial omics images, then we don't need the current standard. We can actually do this in 3D, and we can do this over time and understand really the omics behavior happening in cells, again, and tissues with these label-free approaches. Okay, so moving now to thinking about the clinic. So we're able to take this technology and put this into a cart. This is a portable cart. What you can see on the top of the surface of the cart, there's a little black box. Underneath that black box is an optical window. And so in the operating room, tissue samples would be handed to us. We would place it on the optical window, would cover the box to keep out room light. And we can collect these types of label-free images just within seconds of this in human breast lumpectomy specimen tissues. These are some of the example images we get. This is normal human breast, again, unstained, label-free. Adipocytes are in blue, the collagen in green. You start to see elastin in yellow, which is new here. This is another example, one that's more collagen, a lot of elastin. But I want you to contrast this with the tumor, where now the collagen is deranged and there's multiple different structures there. A lot of this blue and purple cells, which are the tumor cells that we're picking up in the specimen. Here's another example, DCIS, ductal carcinoma in situ. That's a single duct, about 200 microns in diameter. My students like to call this image the tumor galaxy, because if you didn't know that this came from a microscope, you might think it came from a space telescope of a galaxy somewhere away. But you can see typical collagen signatures, TACs, tumor-associated collagen signatures around the duct and some of the changing collagen. So again, something we're seeing on the microscopic scale. Realize this whole image was really only about 1 or 2 millimeters in spatial extent. Okay, those images, how can we use those clinical images now with our AI model for this real-time virtual histology? Now, if we again think of the standard of care, where that lumpectomy specimen comes out, goes through histological processing, anywhere but Mayo, it takes a day, right, to process all of this. There's often viewed by multiple pathologists. Again, it takes time to be able to do that. But what if instead we have our cart in the operating room, and with very minimal processing, that sample is imaged, the margins assessed, it's fed into this model, and we can get this diagnosis just from that process. This saves a huge amount of time, anxiety, perhaps even the need to return to surgery, right, for multiple resections. So that's been our goal. And what we were able to do with the data set that we've collected in vivo, this was on 14 cancer subjects, human subjects, 72 sites on these specimens, 7 healthy subjects from breast reduction surgeries. And we had enough images, over 50,000 images for a training set, validation set, test set, and all these different tiles. And in the end, after that trained model, we were able to get per tile accuracies of 95%, and per slide, if we looked at that entire set on a slide, close to 100% accuracy for identifying cancer. So in a sense, we now have this model to classify these images. And you can see here, no matter what image we feed into this model, we really get this assessment tile by tile. Is that cancer or is that normal? And depending on how big we choose those tile sizes, we can look at these different ROC curves, but very good results in this classification. Now, one of the things that we're always wonder about with AI is people think of it as a black box, and you don't really understand why the model is predicting or giving this outcome. Often what we can do is, because our input data are images, we can look at the images that the model chose to make different decisions. So this is a t-SNE map. It's a way of reducing all the multiple features and dimensions the AI model looked at down to just a few. And fortunately, a lot of these t-SNE maps are just points on this grid, but each point is an image here. And now if we look at what images did the model use to say that this was a true negative, normal tissue, it was the collagen fiber orientation that really indicated that. The false positives, false negatives, these were those elastin fibers, mixed images that had a lot of different structures present. But the images that the model used to say this was true positive, this was truly cancer, were the images that had these extracellular vesicles present, and vesicles that were rich in NADPH. So this really opened up an entire new direction for my lab, because we wanted to better understand how these vesicles and the images that we could generate, how those could be used for diagnostic purposes or screening. So these extracellular vesicles are associated with cancer. They're also present in normal homeostasis as well. But these vesicles bud off or emerge from parent cells in several different ways, and different sizes. So there are the small extracellular vesicles. They have different names. Historically, they were exosomes, really small, 30 to 150 nanometers. Then there's these microvesicles that actually bud off of the membrane from the parent cell. And then there's apoptotic bodies, which I think we're familiar with. But the point being is that when these vesicles carry with them signaling molecules, RNA, DNA, proteins, that cells will use to communicate with other cells across long distances. They have all sorts of surface markers on these, but importantly, when they leave the parent cell, they take with them a bit of the cytoplasm, a bit of that parent cell goes with them in that vesicle. And that's what we use and take advantage of as well, to image and visualize these. So again, it's really been a challenge for the field to work with extracellular vesicles, because generally the tissue is resected, it's fractionated, you separate with ultracentrifugation, you separate out these vesicles and you purify them. But when you do that, you pool them. So you lose the spatial content and context of where those vesicles were. That's what we get when we image. We can see some vesicles in the blood vessels, some are outside of the blood vessels, many different locations. And it's really the SLAM approach that allowed us to be able to see these vesicles label free and get this metabolic signature. So how do we characterize these? If we do purify the vesicles, put them on a microscope slide and image them, they just appear as single little pixel dots. Now, even though our resolution of the system is around 400 nanometers, we can still see vesicles that are smaller than that, because they just show up as we can detect their content and signal. We may not be able to image them spatially, but we know that they're there. So every little pixel you see has a multimodal signature. And then we do EM to verify that, and indeed, we're looking at these. Now, we did a study where in culture, we can extract those vesicles from different cancer cells, different cell types, some that were invasive and very aggressive, others that were not yet invasive, and then normal cell lines. And we use this redox ratio to assess the metabolic signature of the vesicle. Now, what you'll see is that depending on from those different cell lines, we see different histograms, and we see a shift toward a value of that redox ratio toward 0.5 in the more aggressive cell line. So all of a sudden we can get different signatures from vesicles that tell us how aggressive the parent cell was when it produced that vesicle. Okay, well, we can image these in different ways. We can look at those cultures and we can see these cells that are producing these vesicles. as they come out there. In some cases, we can, if you look in the yellow box, we can see a vesicle that is taken up by another cell, and that cell looks like it begins to undergo apoptosis. And here's just a cleaner view of that over time. It starts to bleb. After that, here it comes. And after that vesicle was taken up. So there's a lot of dynamics that we can start to study how these vesicles interact and are produced as well. We can also see how these vesicles are moving through tunneling nanotubes between cancer cells, how this content is packaged. So not only do they get exocytosed and communicate, but they through these nanotubes as well. And if we look more closely at these images, and again, the little blue dots, the tumor cells, you can see evidence of where these vesicles are located. But the reason we detect these is because they're this concentrated NADPH and that autofluorescence. We also detect the structure of the vesicle with this third harmonic technique. So again, a multimodal signature. But we're seeing some surprising things, like the vesicles actually have a higher concentration of NADPH than their parent cells. So is there a concentration mechanism that's happening? Are there fragments of mitochondria that are generating this within the vesicle after it's left the cell? We don't know, but we're seeing some really interesting features of this metabolic signature within these vesicles. So going back, if we look at the rat tissue again, the images, we can extract out where the vesicles are, and we see those vesicles have that same shift. In the cancer tumor tissue, we see that that histogram is more toward 0.5 compared to the control cases. And if we then go back and look at our clinical data, first off, if we just count the number of vesicles, you can see in the human cancer cases, the number of vesicles present in the tissue is statistically significant compared to the healthy breast reduction cases. So just by counting alone, we can look at this. And in also, the number of those vesicles increases with the more aggressive cancers. So again, just a very simple way of sort of assessing the level of aggressiveness of that tumor that's being resected or imaged. Then we've also started looking at the distribution of these. We don't have a lot of final results, but looking at how these vesicles traffic and get move through the tissue. They don't have an inherent mechanism for transport. We do believe macrophages pick them up and transport them systemically. Anyways, coming back to the human tissue, we can look at those human breast tissue images and again extract the vesicles and see that same trend, that a shift in that metabolic signature for the more aggressive cancers. Now, we had only 2 cases of vesicles from a patient that was 2 years out of her breast cancer surgery, no clinical history or evidence of residual disease, but her optical signatures of vesicles shows an aggressive signature. And we don't know if there's underlying aggressive cancer somewhere, or what we hypothesize is that once a body has been preconditioned to host a tumor and metastatic disease, that doesn't revert back. And so these tumor cells will early on produce these EVs, send them off into the circulation. These EVs are taken up by peripheral cells that upregulate their gene expression, their mass production, to really essentially precondition our body to host the tumor and the metastatic cells. So the question, does our body remain preconditioned even after that primary tumor has been removed and treated? And is this why we know that cancer survivors are at risk, greater risk than the general population for secondary malignancies? We think it's because of the radiation and the chemotherapy, but is it because perhaps they were preconditioned by these extracellular vesicles and their body remains primed like that? How do we reverse that state? But these are some really fundamental questions we're starting to explore with this technology. So across the gamut, we've looked at all kinds of different tissues. We're seeing things people haven't seen before with these. And so it's just been an incredible journey to look at the different tissue types, look at some of these contrasts, trying to figure out what we're seeing in these structures, and are we seeing things that could potentially be new biomarkers? Now, in addition, it's this concept of the living biopsy. So we spend all this effort to biopsy tissue, remove that piece of tissue from, say, the liver or the lung or the breast, one of the first things we do when we get it is we plunk it into formalin and we kill it. And we kill that to preserve the structure and be able to look at that structure for diagnostic purposes. But what if we took that specimen and we cultured it for even 24 hours, right? We can culture tissues and even organs, rat organs for days. Can we actually culture this? And during the time that's being cultured, image it label-free like this to better understand the cell dynamics, the metabolism, how are those cells and that tissue really dynamically moving? Organoids, spheroids are filling this void. But we think this living biopsy concept can open up so many new directions and diagnostic information. So just to conclude, these label-free techniques really have the potential to change, when, where, and how we assess tissue microscopically. This can lead to stain-free, label-free histopathology coupled with AI to deep profile this environment. It leads to new biomarkers like these extracellular vesicles. And if we think about how this technology now can be integrated into other platforms at the point of care or at the point of procedure, like even greater precision in surgical interventions. If we think about surgery historically, you can see examples here, right? Surgery has advanced in so many technological ways. I like to say, well, now we have color, right? Back from the black and white photos, but the surgeons are still, approaching, looking at the patient and the surgical site in the same way. Maybe they're using their eyes, and maybe they're magnifying their vision, right, to magnify the surface features. Or maybe we have these really advanced technologies, intraoperative MRI or ultrasound, to be able to look at that tissue during the procedure. However, none of it is looking microscopically at what's going on within that surgical site. Instead, we send the tissue to pathology. So in the case that I've just went through in breast cancer, if we can really bring in technologies intraoperatively to image, think about the reduction in reoperation rates that may be possible and just incredible savings in cost and anxiety to patients. So this is what led to our interest, really our joint interest through our Mayo Clinic and Illinois Alliance for Technology-Based Healthcare to put together teams to respond to an ARPA-H call, where ARPA-H was asking, what if surgeries fix problems flawlessly the first time? And it seemed like it was ideally suited for this technology, where we wanted to put together and build this Margin DX, margin diagnostics, to be able to assess in real time, label-free, these margins during surgery and make sure that the surgeon removed all the cancer tissue. So this was, like I said, a very large collaborative project between Mayo Clinic and the University of Illinois in Urbana through our alliance with multiple teams at Mayo here and at Illinois, also our National Center for Supercomputing Applications, Carle Health, a startup, our cancer centers, really a large 75+ person effort between our institutions to address this challenge. And what are we doing? Well, it's coupling and designing a technology where we can use a label-free imaging, first called optical coherence tomography. I didn't talk about this one today, but think of it as the optical analog to ultrasound, but very fast. And we can image with this probe the larger tumor cavity and identify areas that are suspicious. Then use this high-resolution nonlinear imaging coupled with the AI to be able to identify where these positive margins are. This addresses what I call a multi-scale challenge because it really is a challenge to image large areas of any tissue at microscopic resolution. And you really have to bring in multiple modalities to do this. So that workflow, we use OCT with a handheld probe, and that probe, then the surgeon will sweep that across the tumor cavity surface, the resection bed, after the tumor has been removed. The system tracks where that probe is located, and AI is analyzing that incoming real-time data and flagging areas that are suspicious within that cavity. We then have a robotic arm with the nonlinear optics to go back to each of those sites and generate that label-free digital histopathology with AI to determine, is that cancer or not? And so with this, not only in these open procedures, but with these technologies, we can design this around probes, laparoscopic robotic surgeries, flexible endoscopes, but we think there's a huge potential to using these label-free techniques for many different procedures in medicine going forward. So I just want to close here and thank my own research group at Illinois, a wonderful group, a group at many different career stages, from undergrads to grad students, research scientists, a lot of collaborators and postdocs, also funding from many different sources that really contribute to this interdisciplinary approach. And thank you very much for your attention. Happy to take questions. Questions always lead to new ideas. Thank you.
I'm manning the microphone, so if there are questions, if people want to raise their hand, I'm happy to bring the mic up to you. Here we go.
Really fantastic talk and interesting images and things. I'm curious if you've thought of applying, you kind of implied this from the latter slides and things, how this might be adapted, let's say, for example, to issues associated with the kidney, like kidney stones and things, which are sort of uniquely suited for having, edges and barriers. And I mean, they affect 1 in 11 people in the US, so much more frequent than cancer even. And even for doing things like liquid biopsies, have you pursued that? And
Yeah, no, a lot of good questions too. So first off, accessibility. A lot of the engineering that we do is engineering these beam delivery systems. So with these optical techniques, we can feed this light down optical fibers. We put these into needles, into catheters, into endoscopes. So anywhere you can pretty much snake a catheter, you can actually deliver this type of light and kind of do this type of imaging. There's sacrifices in terms of the quality, of course, but there's that accessibility. Liquid biopsy. So we actually have started to look at extracellular vesicles in urine. And that's a bit of a challenge too. We can isolate these EVs. We can look at their signatures. The problem with urine EVs is that we kind of don't always know where they came from. And there's a lot of other, we believe, other conditions that might affect the signature within those urine EVs. But if we could identify clear signatures, say as a screen for cancer, since we can distinctly differentiate based on metabolic activity, that's a much easier screening. Everybody has to pee, and so we can screen easily that way. And I'm sure there's many other ways that stones and the presence of those can be recorded from liquid biopsies of urine, or I don't know, even in the blood as well. But yeah, I think that we've been able to extract EVs from blood, from serum, from urine, many other sources. They're kind of everywhere, but it's figuring out the signatures that is a little bit difficult.
In the back.
Hello. Okay. Fascinating talk. I think the imaging with the EVs is fantastic. I just had a quick question about that. Are you able to distinguish between looking at extracellular vesicles versus looking at lipoproteins, which, as you know, have very similar size and lipids and everything? Are you able to decipher between them in your imaging?
Yeah, good question. So from this multimodal signature, yes, we believe we can see those differences. One of the other label-free techniques I didn't talk about much here is that CARS, that coherent anti-Stokes Raman scattering. That is very sensitive to lipids, fatty acids, the degree of saturation of those bonds. We can actually look at essentially Raman spectra coming from each of these vesicles. And so an extracellular vesicle, given its cargo, also maybe given a lot of the corona or halo that might be around on the exterior of that looks very different from, say, a lipid droplet or purely lipid vesicle. That said, I think there's still a lot to understand there. Our technique really is looking more at this metabolic signature within the EV. We aren't able, and I think this is a question in the chat too, we're not necessarily able to look at the content, the DNA or RNA or protein content within the vesicle itself. That's something we're trying to think about how we could do. But right now it's just this The signatures we get back from the vesicles are this metabolic signature and sort of the size or shape of the vesicle itself.
Over here on the right.
Fantastic talk. Thank you. I'm a vascular neurologist, also a trialist. So I'm thinking of your work in terms of clinical applications, particularly for, true physiologic endpoints for interventions. Can you please comment on applications for non-invasive monitoring, particularly for CNS structures, vascular, and how that might actually play out in real life? And then the second question is the scale. We're looking at like angiograms, we're looking at like 1 millimeter resolution, and what might be learned at maybe zooming out a little bit and kind of discuss some of those implications.
Sure. Yeah. So, one of the challenges we always face, or there's a trade-off that we make. Not only I talked about the multi-scale challenge, but also the fact that we sort of sacrifice depth of imaging for resolution. And so we're not able with these optical techniques to image very deep in tissue. In fact, we can only go, up to 1 millimeter at most. And so that's where a lot of this engineering of how do we get the light to the tissue. And once we're there, we can do this high resolution imaging. From the vascular perspective and clinically, so if we image the skin, we can actually see kind of an in vivo flow cytometry. We can get deep enough into the superficial kind of papillary dermis where we can start to see some of these loops, these capillary loops, and see individual red and white blood cells going through these capillary loops. That's one area that we can access the microvasculature. The eye is another as well. So we have OCT, in fact, is used quite commonly to look at the vasculature, angiography in the eye as well. But we can have access to the vasculature there as well. Even like on the lip, the exposed lip is another site. But we have to kind of look strategically of how we can get access to that vasculature. And as much as we can maybe tell from those sites, maybe there's markers.
Another question up here.
Okay.
Hello, I work in OCTs as well. So nice presentation. Like to see label-free technique being shown quite extensively like this. So my question is, have you looked at inelastic scattering, maybe Brilliant microscopy also with respect to other imaging modalities when you looked at tissue? The question is specifically, have you looked at biomechanics as well in comparison with your original work?
Yeah. So thinking about this optical elastography technique. So I didn't talk much. My lab does about half nonlinear, half OCT. So there's a whole nother aspect of, in fact, I trained under Jim Fujimoto at MIT, who developed OCT and was part of the early developers of that. But there is this use, any of these techniques can be used to look at the sort of the mechanics. And with OCT, we haven't looked at the Brilliant type microscopy techniques. But with OCT or any of these fast modalities, when you apply strain or compression or movement, right, you can begin to assess the mechanical properties of the tissue. One example I can say we looked at the skin, and a lot of the mechanics, tissue mechanics, is either at the single cell level where they're perturbing a single cell and looking at its mechanical properties, or it's the whole tissue level. But there's not a lot of work done at mechanics of in between those scales. So what we did is we looked at imaging with this multiphoton technique, different layers in the human skin, as we applied kind of a stretch, right, to that skin. And what we could see is that the cells will actually, you would think the cells at all layers would just follow that direction of that strain, but they don't. So depending on the structures, those cells are actually moving in different directions in the sheet under those forces. So it's a way of better kind of understanding at the microscopic scale what the cell mechanics are in tissue, in situ. And I think there's a lot more that can be done on that scale too. But it is interesting. Like we've been trying to look at straining cells and seeing how the metabolism changes, for example, since we have access to this label-free metabolic signatures, but a lot more to do in that space.
So my disclaimer is I'm a primary care doctor, for those of you who don't know, but the most obvious application of this that comes to mind is my elderly population have multiple skin cancers.
Yeah.
And Mohs surgery is a very common condition for many of my patients, which is often hours and hours long
Yeah.
as they go back and resample, look for microscopic advancement of tumor. And it would seem like this would save hours of time were it in the derm suite where they're doing the derm surgery. Has that been looked at?
So absolutely, right on in terms of the application, the need there as well, and a good match. So not so much this nonlinear technique, but people have used OCT or reflectance confocal, it's called. So just a reflected endogenous signature that can get at microscopic resolution to be able to look at those patterns during Mohs surgeries. And you're absolutely right. I think anywhere where we use histology now, there's a potential to use this label-free at the point of care, right? At the point of procedure. So many opportunities there. I think one thing too, me being trained in internal medicine, primary care, a lot of what we've been trying to do is advance the technology, closer to that first encounter. And if you think about, we spend a lot of time in what we call primary care imaging and how we can take these technologies. And if you look in the primary care suite or in the ER, right, the optical instruments that are there are the otoscope and the ophthalmoscope for looking into the eye or the skin, right, or the ear. And that again is just light that's kind of magnified view, right? We say it's a pen light with a magnifying lens. But with this technique, we can see below the surface. So we've been able to look at ear infections, otitis media. We can see behind the tympanic membrane, see if there's biofilm, see if there's an effusion, really improve the diagnostic capabilities of the primary care doctor. We've been able to look at, in the oral cavity for suspicious lesions, the skin, right, for screening. The skin is really interesting because if it's a single lesion, is it a dysplastic nevi? Is it melanoma? A single lesion is so much easier to physically biopsy, rather than bring in a big cart and point at it. So, but when you've got multiple lesions, then this technology seems to be a little bit more practical. So, there are a few here, maybe online I can mention. One is, can this technology quantitatively measure expression or levels of specific cellular molecular markers? If yes, which parameters? So kind of speaks to my previous answer too. We have with CARS and SRS, we can look at lipids and proteins and those ratios. And if there is a particular marker, or what we've done is if a drug, for example, has a specific signature, we can track that drug and its distribution through the tissue because we know that signature. But in terms of the endogenous molecular marker, that's a little bit out of our reach at this point. Has there been work done using technology in autoimmune conditions, ulcerative colitis, healing and active inflammation? Not directly for us, but again, I think because of quantifying inflammation with these associating that with our metabolic signatures, I think would be a really interesting pursuit. And then finally, what is the current cost of this technology? Do you expect it to be cost effective? So this is a question that ARPA-H is really pressing us as well. We like to say, so the nonlinear imaging that I was showing you, those systems are no more costly than that multiphoton microscope in your core imaging facility, right? A microscope, like roughly about $500,000. However, the information that it gives, we believe, is going to compensate for that, right? And the fact that in maybe a rural clinic or smaller hospital that doesn't have a pathologist on staff or doesn't have that pathology lab, being able to think about the system replacing or augmenting a lot of that type of care, all of us, and also reducing reoperation rates from the current 20% down to maybe less than 2%. Those savings, we believe, can compensate for the cost of one of these instruments. Now, some of the other technologies, OCT is very low cost. And in fact, those are inexpensive enough. They are in the primary care clinic. They are in the small rural hospitals. It's just for different applications.
Well, I would like to thank Dr. Bopart for coming today. This has been a fascinating talk, and I think it's stimulated a lot of really good questions, and really is something when we think about innovation and thinking about the future and what it holds for both people like me in primary care and those individuals with oncology and other fields, that there really is a lot to look forward to. And again, Dr. Bopart, thank you so much for coming to Rochester to Mayo Clinic for our Center for Individualized Medicine Grand Rounds. Thank you.